This eruption of colorful foam is a staple of grade school science. It is a rapid physical expansion designed to safely captivate a young audience. But reproducing this exact reaction in an undergraduate quantitative kinetics lab presents a distinct professional paradox. An elementary visual demonstration does not easily translate into a reliable, measurable analytical model. The engine driving the foam is the catalyzed decomposition of hydrogen peroxide. The reaction breaks the peroxide down into water and rapidly expanding oxygen gas. Consumer-grade hydrogen peroxide sits at a 3% concentration. Scaling this reaction for strict data collection requires 30% peroxide, which shifts the baseline from a mild irritant to a highly reactive, hazardous chemical. To drive the reaction forward, chemists have to introduce a catalyst. This creates an immediate optimization problem. The chosen catalyst must strike a precise balance between mathematically reliable data and laboratory safety. The two standard options are potassium iodide, an inorganic compound, and catalase, a biological enzyme found in common baker's yeast. Optimizing this experiment forces researchers to weigh two opposing methodologies. They must choose between the highly predictable yet toxic kinetics of inorganic iodides or the safe, statistically complex behavior of living biological enzymes. The potassium iodide pathway operates through a rigid two-step mechanism. The iodide ion acts as a true inorganic catalyst. It enters the reaction to create a temporary intermediate, and in the second step, it forces the release of oxygen gas while regenerating the original iodide ion entirely. Because this two-step process is purely chemical, it is mathematically clean. Students can track the kinetic rate with high precision and near-perfect repeatability. However, examining the formal job hazard analysis for this pathway reveals the steep cost of that precision. This hazard analysis outlines requirements for scaling the experiment. Step 1 and Step 3 explicitly require 30% hydrogen peroxide, functioning as a strong oxidizer. At this concentration, dermal contact results in chemical blistering, and inhalation of vapors can trigger respiratory edema. Introducing the potassium iodide powder into this already volatile environment compounds the risk. Step 6 highlights the risk of inhaling the Ki dust itself. Furthermore, combining this dust with the strong oxidizer generates an exothermic reaction, rapidly expelling hot oxygen gas and creating a serious flammability hazard. The reaction also generates volatile iodine gas as a toxic byproduct, forcing strict disposal protocols. Potassium iodide yields flawless data for kinetic modeling. But harnessing it requires safety shields, chemical fume hoods, and hazardous waste containment, making it restrictive for open-bench educational environments. The primary alternative replaces the inorganic chemical with Saccharomyces cerevisiae, common baker's yeast. The active agent here is catalase. In living systems, this enzyme exists specifically to degrade reactive oxygen species before they damage the cell. Catalase is exceptionally efficient. It reacts more than 1 million times faster than the iodide ion. The mechanism is also far more direct. Catalase drives the decomposition in a single step, entirely avoiding the toxic intermediate compounds produced by the Ki pathway. The resulting byproducts are entirely non-toxic, just water and oxygen. This diagram illustrates the necessary experimental setup to capture quantitative measurements using the enzyme. Because the catalyst is biological, it demands strict environmental constraints. The water must be heated to exactly 50 degrees Celsius. The solution's pH must also be maintained in a narrow window, resting squarely at 6.5. Living organisms are fragile. If the temperature spikes or the pH drifts, the catalase enzyme denatures. Its physical structure breaks down, and all catalytic activity halts. Utilizing yeast eliminates the toxicity and the hazardous waste of the Ki method, but it forces researchers to manage the extreme fragility of a biological system. So how does the fragility of a living enzyme impact the data collected on the lab bench? This scatter plot maps the baseline kinetics, measuring hydrogen peroxide concentration against the reaction rate. The baseline behaves exactly according to standard kinetic theory. Increasing the peroxide concentration yields a tight, predictable linear increase, reflected by the high R-squared value. This second chart maps the catalyst variable. As the yeast concentration increases, the reaction rate scales up linearly. But notice the severe vertical separation. The data fractures into 3 entirely different trajectories, depending on the specific source of yeast used. This fracturing is the direct result of biological degradation. Differences in the age of the yeast and the health of the microscopic population destroy enzymatic consistency from one batch to the next. Grouping the lumped reaction rates into box plots reveals how biological variance breaks standard statistical modeling. The medians are highly unpredictable. Yeast 2 shows a tight, compact distribution, while yeast 1 and yeast 3 exhibit massive variance and different output ranges. The resulting data is heavily skewed. It cannot be accurately plotted on a standard symmetrical bell curve. To accurately model this specific type of fractured biochemical variance, researchers must discard the normal distribution and apply a right-skewed gamma distribution. Replacing the inorganic chemical with a safe biological catalyst removes the physical hazard from the benchtop, but it transfers the burden directly onto the math, forcing students to navigate complex, non-normal distributions. Bringing both pathways together clarifies the systemic trade-off facing laboratory coordinators. We map this choice by weighing safety against kinetic predictability. Potassium iodide secures pristine data, but requires extensive controls to mitigate toxic iodine and oxygen. Catalase allows safe open bench experiments, but demands advanced statistical modeling to track enzymatic degradation. Neither catalyst is incorrect. The choice dictates the core lesson of the lab: mastering hazard protocol compliance or mastering complex statistical variance. The reaction is not a monolith. Scaling it from a classroom demonstration to a quantitative laboratory exercise changes the rules based entirely on the catalyst selected. True chemical mastery does not come from generating the largest physical reaction. It is built entirely on a rigorous, quantitative understanding of the precise mechanisms, statistics, and safety variables driving the process.